Those of you who are here right now, the plan is just to wait a couple of minutes for people to trickle in, and then we'll get started. All right, so today we are going over DNA replication, and my name is Nikhil, and feel free to type any questions in the chat or unmute yourself to ask any. So we are doing this through MCAT 101, so feel free to take these emails down and email either of us with any questions you have. And if you'd like to get on the newsletter or listserv, just send me your email in the chat and I'll get you added. So just before we go into DNA replication, I do want to cover some of the basics and the background, starting with the cell cycle. So there are a couple of phases to the cell cycle. Usually there's G1, S, G2, and mitosis, with some people considering cytokinesis to be a separate phase as well. I'll go through each one. So after a cell divides, we have the first phase, G1, where the cell focuses on growing in size, creating energy and using it, and creating proteins like ribosomes and the endoplasmic reticulum for further development. Then we have the S phase, which is what we're going to focus on for the majority of this topic session, which is where the DNA within the cell replicates. Finally, we have the G2 phase, and this is where the cell does some quality control to make sure everything's still working and that it's ready to finally divide. So it checks that the size is right, all of the materials are available for two daughter cells. And finally, we go into mitosis, which is where the DNA in the cell and the organelles and cytoplasm all divide into two separate daughter cells. Cytokinesis is just the little part where the cells are finally splitting physically apart the cytoplasm and the organelles and reforming their own membranes. So the first three phases here, G1, S, and G2, are considered part of interphase, which is where the cell is doing its normal processes. However, some cells also go into a subset of G1 called G0, where these cells aren't designed to replicate and they will just continue their normal functions. Some examples of these cells are stem cells and nerve cells. So on this graph here or figure, you see some checkpoints as well. I'll go through each of these checkpoints. These checkpoints are also known as restriction points because if the cell is not able to pass all the checks at these checkpoints, it may drop back into G0 or it may stay in the phase before it to make sure that it has everything before progressing past that checkpoint. So the first checkpoint is here between G1 and S. And what it checks for there is that the cell has the proper DNA needed for replication and mitosis, along with the signals and machinery for replication. This is really controlled by a protein called P53. And if it doesn't pass this checkpoint, the cell will try to repair its DNA. The second checkpoint here between G2 and S really happens some part through the G2 phase, where the cell checks that the DNA has gone through proper duplications, that there are no mistakes, and it has the required size organelles and cytoplasm to divide properly into two separate daughter cells. And this is also regulated by the protein P53. Finally, the mitosis checkpoint. This happens partway through mitosis, where the cell ensures that the chromosomes are attached properly to the spindle plate, which is the plate that they align at before dividing into two daughter cells. And this whole process, the cell cycle, is just regulated by a cycle of proteins called CDKs and cyclins. And last thing I wanted to cover in terms of basics is just DNA structures, because that's what we'll be going over later. So we start with a nucleoside and a nucleotide. Nucleosides are 5 carbons in a ring along with a nitrogenous base, which you can see in the structure at the bottom right, where you have the gray 5-sided sugar and the colored nitrogenous base. A nucleotide is the carbon, the base, and a phosphate group attached. And each nucleotide is laid out in a chain and bonded to create one strand of DNA. There are 2 types of nucleic acids, and that's deoxyribonucleic acids and ribonucleic acids. The main difference that may come up for you on the MCAT or just in general is that on the 2' carbon of the sugar, the gray molecule here, the ribonucleic acids will have a hydroxyl group, an OH, and a deoxyribonucleic acid will have an H, just a hydrogen. So then we get to the backbone of DNA, which connects the carbon and nitrogenous bases. And this is done through the sugar and the phosphates. From the 3' carbon of the sugar to the 5' of the phosphate, there are bonds formed between each of the phosphates and the sugars, and these bonds are called phosphodiester bonds. Now, as I'm sure that you've all heard, there is base pairing between the 4 main nitrogenous bases used in DNA, and the number of bonds differ based on the bases being used. And between adenine and thymine, there are 2 hydrogen bonds, and between guanine and cytosine, there are 3 hydrogen bonds. Now, an easy way I like to use to remember the difference between the bases, as in purines and pyrimidines, is cut the pi, and pi being pyrimidines and cut being C, U, and T, which are cytosine, uracil, and thymine, respectively, being pyrimidines, and adenine and guanine being purines. Purines also have 2 rings, as you can see in the image at the bottom right, while pyrimidines, as in C and T for DNA, have 1 ring. Now, usually DNA exists in double strand configuration, where each strand is anti-parallel, and they are read from the 5' of one end to the 3' of the other end. So one strand from top to bottom will read 5' to 3', and the other strand from top to bottom will be the opposite, 3' to 5'. When you have DNA bonded together in dual-stranded configurations, you normally have a double helix, which you can see in the image at the top right, where they are twisted around each other. And this in nature normally happens as bDNA, which is right-handed, which means it twists up and to the right. And just a couple numbers, it has about 10 bases till it turns all the way around. And a major and minor groove just based on the configuration, and about 3.5 nanometers per turn. Now, another configuration, which is really hard to find in nature just because it's so unstable, is zDNA, which turns to the left. And it's a little bit larger in that it takes 12 bases per turn and around 4.6 nanometers. All right, I have a question. How do you remember the nucleotides themselves? In terms of the actual rings, it's just purines have 2 rings and pyrimidines have 1. I just remember them as being the opposite of the size of the name. Purine is a smaller name, but it has 2 rings. And pyrimidine is a long name, but it has 1 ring. Actually not sure of a mnemonic for a ketone versus an imine. Sorry about that. All right. So histones and nucleosomes. So these strands of DNA are, there's a lot of DNA. Let's start with this. There's a lot of DNA in each chromosome. And to make it tightly packed in a nice configurations, there are histones, which are proteins that associate with DNA inside the nucleus. And the DNA that we have in eukaryotes are associated with 5 different histone proteins. We have H2A, H2B, H3, and H4, which form a nice spherical complex, like you can see in the image at the top right. And they each have about 200 base pairs around a histone. Now, this histone is packed, these histone proteins are packed into even larger complexes called nucleosomes, which basically consists of several histones grouped together with DNA wrapped around them. And the nucleosomes are packed and sealed by the histone protein H1 that sits at the end of each nucleosome and makes sure that the DNA is tightly coiled around each nucleosome. Now you may be wondering, since DNA is packed so tightly, how can our cells express the DNA into RNA and then proteins? There's generally 2 types of chromatin that exists in the chromosome, and that's heterochromatin and euchromatin. What this describes is how tightly the DNA is packed. Euchromatin is where the space between the nucleosomes are spread out, which allows access to ribosomes and mRNA to be made. And this shows up when you analyze DNA as a light color, where you see the chromosomes and you'll see light patches showing that the nucleosomes are spread out and the DNA is uncondensed and it's able to be expressed. On the other hand, we have heterochromatin. This is where the nucleosomes, as you can see in the left side of the top right image, the nucleosomes are packed super tightly and there's really no space for RNA to be made and for ribosomes to translate that RNA. And this is what happens, or this is what shows up as dark colored in DNA imaging. And this just shows that the DNA is super condensed and it is silenced as in not able to be expressed. Then when you look at linear chromosomes, you have 2 things called centromeres and telomeres in eukaryotes, while some bacteria have telomeres. Let's start with the centromere. So for the centromere on a chromosome, in this chromosome, you will see a center point. And this area is heterochromatin, which has high GC proportions just because the G and C form stronger bonds because they form 3 bonds when opposed to adenine and thymine, which form 2 hydrogen bonds. And the high proportion of GNC just makes sure that it's bonded super tightly, and the GC sequence is generally repeated, called tandem repeats, and that allows for noticeable indentations in the centromere area where the sister chromatids can bond together, or chromosomes can bond together. And this is where they are bonded together until anaphase, where they're finally pulled apart by the spindle fibers. And the telomeres, on the other hand, are at the end of the chromosomes. And these telomeres usually start with a sequence of TTAGGG in terms of the base pairs. And they're only there because there are linear chromosomes in eukaryotes. What that means is when you replicate them, as we'll see later, you can't replicate the entire thing, and so you'll lose a piece of the chromosome each time. To prevent that, telomeres have repeating sequences also of high GC base pairs that are basically useless and don't code for anything. That way you can cut them off in replication because you can't replicate everything, and the chromosome will still have full functionality. Telomeres, on the other hand, also have the high GC content just because it will create a stronger bond between the two strands of DNA and prevent unraveling at the end of the chromosomes, which is super important. And as we'll talk about later, telomerase, or I'm not sure how to say it, but it's telomerase, but what that does is it's an enzyme that extends telomeres so that you will always have an area of DNA that you can cut from without losing functional genes. And this telomerase exists in greater concentrations in dividing cells just because they divide often and will cut greater portions of telomeres off because of that every time they go through replication cycles. But yeah, that was the background info. So I have some questions for you guys. And for each question, I will give you some time to answer, and just type the answer in chat when you think you have it. Perfect. I am getting a lot of Bs, and that is correct because B DNA is the type of DNA that is commonly found in nature, and Z DNA is the opposite. It's left-handed, and it's not normally found in nature just because it's super unstable to create and synthesize, or to create and just exist. Right. For the next question, which type of chromatin is expressed? All right, perfect. Euchromatin is the one that is expressed, and it will show up as light colored or bright in light microscopy, while heterochromatin is transcriptionally silent, and it will show up as dark under light microscopy. For the next question, we have which histone proteins seals and adds stability to the nucleosome? All right, so this one is the histone protein H1. And that's just because the other four proteins, H2A, H2B, H3, and H4, just form the circular complex that we see in this image here, where you can see the histones. The H2A, H2B, H3, and H4 proteins form this circular complex. And the H1 protein just makes sure that the DNA is wrapped tightly around each of these proteins. It adds this stability to keep them tightly bonded, just because of the negative charge of the DNA and the positive charges on the histone. And it also seals it by bonding right at the end. Yeah. H1, that is what I consider wrapped, as in it just makes sure that everything is tightly packed together. And for the last question here, which nucleic acids are considered pyrimidines? All right, so I'm getting a lot of C's, and that is correct. Thymine, cytosine, and uracil are considered pyrimidines, while adenine and guanine are purines, which have 2 rings. All right, so now we're going to talk about eukaryotic DNA replication. So first, I want to start with the origin of replication. So first, we have the replisome, or the replication complex, which is a complex of proteins that really just aid DNA polymerase in replicating the DNA. And the origin of replication is the original spot in the chromosome where the DNA strands are broken apart so that replication can start. The replication fork is another term, which is where after the replication process starts. The fork is the area in which the DNA strands are continuing to be broken apart in order to continue replication. Now we have the sister chromatids and the centromere. So when you have a chromatid, it is the copy that you get from your parents. When your cell replicates the copy it gets from its parents, since you have two exact copies, each one is called a sister chromatid. And these sister chromatids are connected at the centromere throughout the process of replication. And relative to bacterial DNA replication, since eukaryotes generally have a larger chromosome, this process will take a lot longer and it's slow. So we generally tend to use many origins of replication in replicating the chromosomes. And that way, and so we also have many replication forks. Now we have the initiation of replication. So supercoiling is just a type of coiling in the DNA where it is coiled super tightly to pack it closer together. And this just assists in packing the DNA even tighter so that the chromosomes can be super condensed and not take up too much space in the cell. And this is really important when you have to replicate, because when you unwind the DNA, you end up introducing extra coiling further down. And that's what helicase does. It unwinds the DNA from the double helix that we talked about earlier so that the strands can be separated and DNA polymerase can bind to the DNA and do its thing. So the helicase just unwinds the DNA and opens it up and creates more supercoiling or positive supercoiling further down the strand. Then what happens is we get single-stranded DNA binding proteins, which bind to the DNA and really just prevent nucleases in the nucleus from degrading the DNA, because any free DNA is normally degraded by these enzymes called nucleases. It also prevents, or single-stranded binding proteins also prevent the DNA strands from associating with each other again and preventing DNA replication. And we also have semi-conservative replication of the strands. Since we're keeping one parent strand using the single-stranded binding proteins, and we're replicating each of the strands separately, half of the new DNA double helix will be the original parent strand in blue in this image here, and the other half will be in yellow or orange in this image here. And that's what semi-conservative replication is. It means you keep half of the original and have half new. All right, another enzyme is topoisomerase, which really helps to relax the DNA ahead of the replication fork, because as we mentioned earlier, helicase adds coiling to the DNA, positive supercoiling to the DNA further down when it unwinds the DNA for replication. And what topoisomerase does is it cuts the DNA and then lets the tension relax a little and then reseals the DNA back together so that there isn't so much tension from coiling further down the DNA strand that'll prevent DNA replication later. Then we have RNA primase. And what this does is, since DNA can't be directly bonded without a 3' to be bonded to, and that's just a property of DNA, it can't be directly added without something to attach to, primase uses RNA and adds about 10 base pairs of RNA to be added to the strand, as you can see in the image, and that way the DNA can build off of that. And we'll talk about this a little bit later in terms of the leading and lagging strands. What happens is on one side, you'll have just 1 or 2 primers, and on the other side, since DNA has to be built in a specific way, you'll have to continually add RNA primers and build backwards. And that's the lagging strand that you'll have to do that too. All right, so we have DNA synthesis. So how this works is there are 3 different DNA polymerases that are the main participants in DNA replication, and that's polymerase alpha, polymerase epsilon, and polymerase gamma. Or I believe, God, I'm not the best with Greek letters. But the polymerase read 3' to 5', which means they read in the direction that the new strand is going to be built. So if you look at the, let's say, the lagging strand here, and you have the 5' to 3'. So the 5' would be on the right of the lagging strand here, and the 3' would be on the left here. Since you have to build the strand in an anti-parallel fashion, which means going from left to right, since the original strand goes from right to left, the DNA polymerase also has to read from left to right, which means it reads from 3' to 5' on the original strand, but it adds nucleotides from 5' to 3'. And it adds nucleotides that it finds in the nucleus, as in deoxy ATP, deoxy CTP, and so on. And it breaks the pyrophosphate or 2 of the 3 phosphates attached to these nucleotides off when it adds the nucleotide to the DNA strand. And when it creates the phosphodiester bond between the existing RNA primer and the nucleotides that it's adding, it releases pyrophosphate. And then we come to the leading and lagging strands, where one strand, since it's open and is going in the direction that the replication fork is moving, can be continuously copied. So all it needs is one primer, and then the polymerase can just continue adding on nucleotides as the replication fork progresses. While on the other strand, the lagging strand, since it's building away from the replication fork, it'll start, continue for a little bit, and then have to stop and start over at the spot that the replication fork has newly opened up. And since you have to stop and start over so many times, there are little fragments of the newly added or newly synthesized DNA that is created called Okazaki fragments. And you'll see that in the lagging strand here in the image as the little pieces of the RNA primer and the sequence of DNA nucleotides here. Then we have the sliding clamp, which is generally associated with each of the DNA polymerases, just so that it can keep the DNA polymerase more tightly attached to the DNA and so that it doesn't fall off. Hold on 1 second. All right. So we have DNA polymerase gamma, which is the symbol that looks like a Y. And this is also important because it replicates mitochondrial DNA in eukaryotes, because these mitochondria are super important for being properly split up in each cell, because without the mitochondria, you're not going to get the energy that you need for the cell to do its normal processes. And this sliding clamp that we talked about earlier, it associates with the DNA polymerase delta and the DNA polymerase epsilon. Yes, okay, these DNA polymerases in numbers, actually there are 2 types of DNA polymerases, and you'll see that when we get to the bacterial DNA polymerases. And the bacterial DNA polymerases are the ones that actually use numbers. The eukaryotic ones tend to use letters like alpha and epsilon and delta and beta and gamma. Does that make sense? And yeah, guys, let me know if you guys have any questions. Yeah, perfect. All right. So after we get the DNA synthesis of the RNA primer and the Okazaki fragments and the continuous leading strand being replicated, we have DNA editing. And that's just because we have some things that we can't exactly have in order to have properly replicated DNA strands, things like RNA primers. So in eukaryotes, what happens is there is an enzyme called RNase H, and that removes the RNA primer once it's been added and the Okazaki fragments further ahead have been created. Now, once the RNA primer is removed, what you end up with is little gaps between the strands of DNA, as you can see in the image. And what happens is polymerase delta seals that up by adding the DNA, since we now have a backbone of new strands of DNA to work off of. Now that we have all the nucleotides filled in, you still need to connect these Okazaki fragments together. And that's where DNA ligase comes in. It seals the backbone, the sugar phosphate backbone of these separate Okazaki fragments and anything on the leading strand needing that together, especially after proofreading. And that way you have one continuous strand. Now that we know how the normal strand has been created, for eukaryotes, we also have telomerase. What that does is it binds to a specific sequence that it recognizes in the telomere and then extends the DNA strand by adding on high levels of G and C onto the end. And what usually happens after replication, just because as you see with this lagging strand, you can't always replicate everything because of the little gaps, is you'll end up at the end of DNA strands at the telomeres with a little overhang, as you can see in this bottom image, where there is an unpaired section of DNA. That's the section that telomerase recognizes and adds extra nucleotides onto so that it can extend telomeres for more cycles of replication. All right, so that is eukaryotic DNA replication. Does anyone have any questions so far? If so, please unmute yourself to let me know or type it in the chat. In the meantime, I do have some questions just to make sure you guys properly understand eukaryotic DNA replication. So which type of supercoiling do helicase and topoisomerase induce? All right. In this situation, supercoiling induced by helicase is actually positive because as it unwinds DNA at the replication forks, it actually winds it tighter further down. It's really hard to think of a practical application where you can see that, but it's kind of like twisting a piece of string so many times that it like wraps up super tight when you try to unravel it at one end. Yes, exactly. It's like a phone cord. When, let's say, if you guys ever remember like these super old telephones with like the wrapped like circular, I guess, helical cords, If you ever tried to grab a point in the middle and stretch it open so it was straight for a section, the parts above and below it would coil up super tight. And that's what helicase does. When it unwinds and opens up the DNA, it makes the parts ahead super tight. And so it actually induces positive supercoiling. On the other hand, topoisomerase tries to relax that positive supercoiling, and it cuts the strands so that it can relax the tension and then joins them back together. So topoisomerase actually induces negative supercoiling. So in this case, the answer would be B. Please let me know if you need a little bit of a further explanation on that.
So you're saying that helicase is positive because it was what it does down the downstream, sorry, upstream, downstream, up
Yes, it's
downstream.
downstream. Yep.
Right, but wouldn't it be the opposite if you considered what it was doing the opposite way? So really what you're describing is just relative to which direction you're thinking about it?
It doesn't exactly, so helicase doesn't do negative supercoiling because negative supercoiling is still a form of twisting, but it's the opposite way. So helicase opens it up, and that in terms of further down causes positive supercoiling, but in terms of the replication fork where we're looking at, it's not even inducing negative supercoiling. It is just opening up the strand. It's kind of like screwing something in. Sorry, I'm not sure if the best way to explain it. Does it kind of make sense? I do see what you're saying in that relative, it does seem like it's positive supercoiling when you look at downstream, but when you look at the actual replication fork, it's negative, right?
Yes, thank you.
Yeah, sorry. But in terms of supercoiling, generally we're looking at the whole strand. Any other questions on supercoiling by helicase or topoisomerase? If not, I do have a couple other questions for you guys on eukaryotic DNA replication. So for the next one, which eukaryotic DNA polymerase replicates mitochondrial DNA? All right, perfect. So I am seeing a lot of C's, DNA polymerase gamma, and that is the one that replicates mitochondrial DNA in eukaryotes. As to the other options, we have A, DNA polymerase alpha, that assists in DNA synthesis of the chromosomes. Polymerase 3, yes. Polymerase 3 is the prokaryotic equivalent, which we will get to when we cover prokaryotic DNA replication, which is actually just the next part. So yes. Oh, actually, polymerase 3 isn't for mitochondrial DNA. That's the analog of polymerases alpha, delta, and epsilon. Prokaryotes don't have mitochondria, so there isn't an analog for this DNA polymerase, if that makes more sense.
Would it be too much to ask for you to just briefly go over what each one of them does?
Yeah, that's actually what I was about to do.
Thanks.
Yeah, no worries. I completely understand. It's a lot of letters, and personally, I'm not the best at memorizing Greek letters either. So I will definitely go through all of them. So we have DNA polymerase alpha, which is option A. And DNA polymerase alpha assists in regular DNA synthesis of the chromosome. DNA polymerase beta, which I didn't really discuss here, and that's because it really functions in DNA repair. And that's option B. Option C is DNA polymerase gamma, which replicates mitochondrial DNA in eukaryotes. And because it's specific to mitochondrial DNA, there is no analog in prokaryotes because prokaryotes don't have organelles. The fourth option, D, is DNA polymerase delta. And DNA polymerase delta also functions in chromosomal DNA synthesis, and it is assisted by the sliding clamp, which is formed by the pCNA protein as a trimer. And that all you have to remember about that is it helps the DNA polymerase delta maintain a strong connection with the template strand that it's building the new strand for. Same thing with option E, DNA polymerase epsilon. It also replicates chromosomal DNA, and it is also assisted by the sliding clamp of the pCNA trimer. Let me know if there are any other questions about the different DNA polymerases for eukaryotes. DNA polymerase alpha is assisting in the DNA synthesis of chromosomal DNA or replication, and it is not associated with the sliding clamp or assisted by the sliding clamp. Oh, one more thing.
So
Oh,
you're
sorry.
saying that delta and epsilon are used to replicate chromosomal DNA, but that in alpha it's used to replicate chromosomal DNA, but it doesn't use the sliding clamp. So where exactly is it being used within like the cycle?
All three, as in alpha, delta, and epsilon, all three function in chromosomal DNA. They're just used in not in specific locations, but they all three assist in doing the process. And pretty much for the purposes of the MCAT, they work together in doing it. And it's not like they have specifically different functions other than epsilon, option D, which also assists in filling in the bases that are removed by RNase H. So if we go back a couple slides, you will see that polymerase A or alpha, polymerase epsilon, and polymerase delta all function in replicating chromosomal DNA. And it's just that two of them, delta and epsilon, are assisted by the sliding clamp made out of the pCNA trimer, and polymerase alpha is not. The only special role out of these three is polymerase delta, which also fills in the bases or the gaps that are left when RNA primer is removed by RNase H. Does that make sense?
So the delta is like polymerase one, and then the polymerase three, I guess, is between alpha and epsilon.
Pretty much, yeah. That's actually a really good way to put it. Man, you guys are really taking my slides too early. I haven't even gotten to prokaryotic replication yet, and you already know. But yeah, here is the next question. And for this question, you can assume that we are talking about a human somatic cell. All right, so the answer to this question is actually D, 46 chromosomes and 92 chromatids. And that's because in humans, we normally have 23 sets of chromosomes, and each set is from our mother or our father. So we have actually 46 unique chromosomes. And when you go through synthesis, you're basically replicating each one of these chromosomes to create two sister chromatids. So you have 46 chromosomes and 92 chromatids. Does that make sense to everyone?
I have a question, but is it chromatid the arm of the chromosome, which if they are before the replication, like if you replicate, the number will be double. But if you count the chromosome, because you count by the chromatid, the centromere, so the chromosome will be half of the number after synthesis?
Well, chromosome refers to the unique genetic material, right? So
Yes.
when we get 23 chromosomes from each parent, we have 46 in each cell, right?
Yes.
So once you go through the synthesis, you have the same 46 chromosomes, but they're now doubled. So you have basically twins of each of those chromosomes. So you have 92 chromatids. Since
is this like if this is before the division,
before the
mitotic
division, you'd have 46 chromosomes and 46 chromatids.
But if the centromere does not separate, then are you still counting as like if they have 4 arm or 8 arm, if they are all attached by one centromere, are we still counting as one?
Well, it really depends on just the amount of unique genetic material, right? So when you consider a chromatid, you consider like the 2-armed piece, right?
Yes.
Yeah, that is actually a chromosome before replication.
Yes.
So since we get 46 of those 2-armed pieces and you replicate them and you get 4 arms for each of them, right? And so each of the 2 arms is now considered a chromatid because the unique genetic material is the same across each of the 4, across like both chromatids, right?
Oh.
Does that make a little more sense?
Yes, I was a little bit confused because I thought even though if they have like a multiple arm, as long as they are not separate by the centromere, then we will not
I
count
could
as
try to draw that for you.
Yes.
Sorry, because it might help. So each of the chromatid is like this, right? Sorry for the really bad drawing, but this is what one chromosome is going to be like with the centromere right about here, right? So when you replicate this chromatid or this chromosome, you get 2 sister chromatids with this. Let me change the color real quick. With this being 1/2 and this being the other half, right? So each of these are the sister chromatids. However, since they are still sharing the exact same genetic material, it's still one chromosome.
Oh, I see.
Does that make sense now for you?
That makes much easier. Thank you.
Yeah, no worries. I also struggled with this for a long time, and I struggled with the 2N and the N and what a chromosome and what a chromatid was. So I completely understand.
I don't know why, but I'm just not seeing it. Like I follow the picture, but I think I'm more confused now.
Gotcha. So I'm gonna try to explain it again because I know it definitely took me a while to understand it myself. So we have the 22 pairs and then the X and Y chromosome for a human, right?
All right.
So we get one pair of each of those from our mom and from our dad, right? So now we have 46 chromosomes in 23 pairs. For me, I have an X and a Y, and then the 22 22 pairs of different chromosomes for my mom and dad, right?
Mm-hmm.
So we're basically saying we have 46 different chromosomes that look like this.
I think this is where my confusion is because
Mhm.
I was under the impression that that is a chromatid, but it's not. That's a chromosome on its own.
Exactly. That's a chromosome on its own. And
Okay.
I really hate that in textbooks, we always see them as like the big X.
Yeah.
So yeah, exactly. So this is one chromosome. And after replication, once we get the little centromere in the middle that connects the chromatids like this is when you have two sister chromatids. The way I try to think about it so it makes sense is that the number of chromosomes tells you how much unique genetic material you have in each separate piece.
Okay.
Since both chromatids have the exact same genetic material, it's still one chromosome.
Got it.
Does that clear things up for you a little?
Yeah, I got it.
Perfect.
Thank you.
All right. Yeah, no worries. And yeah, if you do sign up for the session via the website, you don't have to submit separate forms. If you select all the sessions you want to join on one form, we'll know. So no worries about that. But yeah, you do count the centriole for the chromosomes, count by the center of X exactly.
And when you were doing it, I think you were doing it for a haploid cell. So then for a somatic cell, it would actually be the chromosome that would be duplicated.
Yes. I believe if you're going through the meiosis 1, it would be the entire thing that is duplicated. I think it's just easier to go by the haploid, so it makes sense instead of having to go through several X's and replicating all of that. But yeah, this is the last question for this section. And then we have prokaryotic replication and then a little bit of proofreading and we should be good to go. Perfect. I am seeing a lot of these, and that's perfect because it is DNA polymerase delta that fills in the gaps from RNase H, removing the RNA primers in the lagging strand and the leading strand. Anyone has any questions about that, please put it in the chat. But I am going to move forward into prokaryotic replication. And thank God, because Greek letter is so much harder than Roman numerals. All right, so we start with the origin of replication again, but there are a couple things I want to mention about the bacterial chromosome. As you can see in this image, which, oh, there we go, perfect. The bacterial chromosome is actually closed and is a circular double-stranded piece of DNA. What that means is we only need one origin of replication because the replication forks can proceed all the way around, like we can see in the image, and join back together to create a completely new chromosome. So something that's important to remember is that there's only one origin of replication and two replication forks in prokaryotic DNA replication. And it's also easy to make this happen because bacterial chromosomes are much smaller, so the replication can happen much faster than eukaryotic chromosomes. I also want to mention that, as we'll go over later and people have mentioned already, there are slightly different proteins used in the replication complex or the replisome of prokaryotic DNA synthesis. And there also are no sister chromatids or a centromere because the bacterial chromosome is circular. So we start again with the initiation of replication. And again, we have supercoiling and helicase, which does the exact same job as it does in eukaryotic cells. It unwinds and opens DNA at the origin of replication. And in doing that, it creates positive supercoils on both sides of the replication forks. So where the DNA is still tightly bound together on the left side here and the right side here, it induces positive supercoils further down in the chromosome. Then we have the single-stranded DNA binding proteins again, which bind to the open DNA, preventing the nucleases from degrading DNA and from the DNA strands from associating together again. We also have DNA gyrase, which is the main difference in the initiation of replication in prokaryotes. When we have eukaryotic topoisomerase in DNA, we have DNA gyrase in prokaryotes. They do the same job, which is adding negative supercoils ahead of the replication forks by cutting the DNA strands and then resealing them once the tension is released. Their names are different. And this is super important if you ever go into like microbiome or pharmacology, because there are specific drugs that are designed to inhibit DNA gyrase, so bacteria cannot multiply. But everything else is pretty much the same, just like primase, which adds the same RNA primer of around 10 base pairs that can, again, be directly associated with the parent strand because it is RNA and doesn't have to add on to the 3' end of another nucleic acid. Now we have the DNA synthesis. Now, instead of the alpha and the epsilon and the delta, we just have DNA polymerase 3. And this is the main DNA polymerase for prokaryotes, which just, again, reads 3' to 5' along the parent strands, like the DNA polymerase alpha, delta, and epsilon in eukaryotes. And it works 5' to 3', adding the same deoxyribonucleic acids and breaking the inorganic phosphate or pyrophosphate off and creating a new phosphodiester bond with the RNA primer. We have the leading strand being copied continuously with 1 primer. Usually in application or in real life, we do see that there are several primers that are used. However, theoretically, all you need is 1 primer that can be continuously extended as the replication fork extends from helicase opening up the DNA. We also have the sliding clamp now associating with DNA polymerase 3. It's still the same trimer of the pCNA protein, and all it does is it makes sure that DNA polymerase 3 is tightly bonded to the template strand, and so can properly create the new bases. So in the synthesis part of prokaryotic DNA replication, everything is pretty much the same outside of the name of the polymerase. Now we get to the editing of the newly synthesized strand, and that's done by DNA polymerase 1. In eukaryotes, it was done by the DNA polymerase delta, which added bases that were that filled in gaps where the RNA primer used to be. However, In prokaryotes, DNA polymerase 1 can both remove the RNA primers by itself and add DNA to fill that gap in. And so it combines the functions of RNase H in eukaryotes and DNA polymerase delta in eukaryotes. We also have DNA ligase, which connects or ligates the backbones of the DNA strands from the Okazaki fragments and from the leading strand wherever several primers are used. The main thing we have to consider here is that there are no telomeres or telomerase in prokaryotes because the chromosome is circular, and so when it's replicated, it is always completely replicated. There's going to be no ends that are left hanging or can't be replicated. All right, and sorry, one second. I do have a last set of questions for you guys, but this section is pretty short just because there's only a couple differences between prokaryotic and eukaryotic DNA replication. So which enzyme is the prokaryotic analog of topoisomerase? Perfect. This one is actually B, and this is just the specific prokaryotic topoisomerase that is used. And like I mentioned before, it is a really, really strong drug target whenever we use drugs for bacterial infections. This is a big target for it because if we can stop bacterial DNA replication, we can generally stop bacterial division, and that way, all your body has to do is kill the cells that remain in the body. All right, for the next question. Oh, pCNA, perfect. pCNA is the protein that when in a trimer makes up the sliding clamp. So when I mention pCNA, it is the component that makes up this sliding clamp. And that's just made by pCNA in a trimer formation. All right, the next question. Which two eukaryotic enzymes functions does RNA polymerase 1 perform? And there's only a couple slides left, so please stick around. I'll try to speed through them as fast as I can. All right, so this one is actually C, and we can definitely go through the functions of each of these enzymes. So helicase opens up the DNA strand from the double helix and induces positive supercoiling. Topoisomerase is the opposite of helicase. It cuts the strands and reseals them so it can release the tension and creates negative supercoiling. DNA ligase connects the backbones of the newly synthesized DNA pieces, and telomerase extends the telomeres in eukaryotic DNA. Now, the two enzymes that RNA polymerase 1 really replicates is RNase H, which removes the RNA primer that is laid down in synthesis, and DNA polymerase delta, at which adds new bases to fill in the gaps left by RNA primers being removed. Primase is the enzyme that lays down RNA primers, and DNA polymerase alpha is just the eukaryotic enzyme that extends newly synthesized DNA strands. So it would actually be C. It would replicate the RNase H function of removing RNA primers, and the DNA polymerase delta's function of replacing the gaps of the DNA primer with, or the RNA primer with DNA nitrogenous bases or nucleic acids. Does that make sense to everyone?
Yep.
Perfect. And then if this is DNA, did I put RNA polymerase? It is DNA polymerase. Sorry if I said RNA polymerase, my bad. But yeah, the last question is, how many replication forks will a bacterial chromosome have? All right, so the answer to this one is 2, which is B, because a bacterial chromosome is circular. And if we go back and take a look at the bacterial chromosome, there's going to be one origin of replication, which is the specific part or place in the DNA where replication starts, and 2 replication forks. And replication forks are the areas where the bacterial chromosome is newly being opened up. So if you take a look at the image here, oh, it didn't pause it, but you will see there's 2 locations where the DNA is continuously being opened up, at the top here and at the bottom here, while there's only one origin of replication, which is where the DNA is originally opened up. Does that make sense to everyone? Perfect. All right. So there, as you can see by this image, there's definitely a lot of different types of DNA repair, but we're only going to be focusing on the ones that have to do with DNA synthesis. So that's really replication errors here, which has to do with mismatch repair. And that can happen when you put in the wrong base, or don't put in a base, or mismatch the wrong base pairs. So with proofreading, and I apologize if you can hear my fan, sorry, my computer almost died, but there's proofreading, which means the DNA polymerase in eukaryotes and prokaryotes, as they're laying down the new bases, will constantly check for if they've ever put in a wrong base by looking at the number of bonds, which create a lack of stability if the correct numbers aren't there, and the methylation of the strand. So in the image at the top right, you can see that when the polymerase, which is represented by this gray shape, adds in the incorrect base, it will notice the lack of stability because of the incorrect hydrogen bonding between the base pairs, and it will use its own exonuclease activity to remove the nucleotide and then put in the correct one. Now, another way that this can happen is the polymerase checks for the methylation of the bases. So when DNA has existed over several cycles of replication, it will naturally accrue more methylation of the bases. And so what the polymerase will do is it will see that the parent strand's bases are more methylated. And so it will know to use that as a template and replace the, in this case, the thymine that is incorrectly placed, and put in a cytosine instead. Now there's also a mismatch repair, which isn't done directly by the polymerases, and instead is done by a group of proteins called MSH2 and MSH1 in eukaryotes, and MUT-S and MUT-L in prokaryotes. And these are very, very close analogs between prokaryotes and eukaryotes. What happens is a dimer of these two proteins will recognize the incorrect base or a mismatched base, and it will call over another dimer of two other proteins called MLH1 and PMS2. And you don't need to remember this, just for, I guess, extra information. And those two protein dimers will fix the mistake by removing the wrong nucleotide and putting the correct one in, just like the polymerase can. The one thing you also need to remember about proofreading is that errors are more likely in the lagging strand because DNA ligase can't proofread the strands to make sure that they're properly placed before it seals the backbones together. On the other hand, polymerase can. So for the leading strand, since polymerase is the one thing that's connecting the backbone and putting in new bases, it can always check and make sure. While ligase just sees a backbone that isn't connected and connects it, it doesn't check for the proper bases or not. And after that, I have a last set of questions for you guys, and that should be it. Perfect. I am seeing a lot of Bs, and that is correct because the lagging strand does have more errors since ligase automatically connects the backbones without checking the correct base pairing. And so if you'd have to replace the bases, it will take longer and there's more likely to be an error. Here is the next question. All right, perfect. I am seeing a lot of C's, and that is correct. There will be 3 bonds between guanine and cytosine and 2 bonds between adenine and thymine. And phosphodiester bonds are between the backbone, so the sugar and the phosphate, while hydrogen bonds form between the specific bases and base pairing. All right. For the last question. So this is something I didn't exactly touch on, it was just there in the image. DNA methylation actually occurs at the nitrogenous base. And that's just really something you'll have to remember. I do also want to mention that it occurs at the histones, and methylation of the histones actually silences it. It turns euchromatin into heterochromatin. That just might be a useful piece of information, so I'm just putting that out there. But yeah, that is the last question. I do want to mention we are a nonprofit, and any donation will help us cover our website costs, advertising. So please consider donating, and definitely tell all your friends that are pre-meds and hoping to take the MCAT and become a doctor just like we are. But lastly, thank you. If you have any feedback for me or for MCAT 101 in general, please use these emails. And again, If you aren't part of the newsletter, then send me your email in the chat and I will definitely get you added. But thank you guys for being here today. I will stick around for any other questions, but that should be it.
Are you still recording?
Hold on 1 second.
It's just like not important for like a recording. So that's why I was
yeah,
don't want to hold
gotcha.
it off longer.